Hello everyone, welcome back. Imagine a wedding where the wrong partners are being matched. What will happen then? Chaos, right? Now imagine that happening inside your cells every second. In your cells, millions of perfect matches happen every second. And the responsibility of making the correct matches lies with the aminoacyl tRNA synthetase enzyme, the ultimate molecular matchmaker. In the previous presentation, we discussed about charging of tRNA, and the enzyme responsible for the charging of tRNA with amino acid is nothing but aminoacyl tRNA synthetases. So in this episode as well, we'll continue our discussion, but the focus will be more on the enzyme aminoacyl tRNA synthetases, the ultimate molecular matchmakers. Now, to begin with, the function of this enzyme is to join cognate tRNAs with their cognate amino acid. Therefore, we are calling them as matchmakers. Now, there are some peculiar features of this enzyme which you need to understand to visualize the beauty of this reaction which they carry out. An aminoacyl tRNA synthetase, an enzyme that attaches appropriate amino acid onto its corresponding tRNA. It is also called tRNA ligase. Okay. Aminoacyl tRNA synthetases are those enzymes which ensure proper amino acids are used to build proteins. Because if they do a wrong charging, there is nothing that can be checked by this tRNA or the other translation machinery to correct the amino acid. These enzymes charge each tRNA with the proper specific amino acid, thus allowing each tRNA to make the proper translation from the genetic code of the DNA into amino acid code of the proteins. Most cells make 20 different aminoacyl tRNA synthetases, one for each type of amino acid. These 20 enzymes are widely different from each other. Each enzyme is optimized for its own particular amino acid and the set of tRNAs that represent that amino acid. These enzymes have very high fidelity. Aminoacyl tRNA synthetases must perform their task with ultimate accuracy. Every mistake they make results in a misplaced amino acid at the protein level. Therefore, these enzymes make about one mistake in 10,000 charging events. You can see how specific they are. In 10,000 charging events, they are making only one mistake. Let us first understand how do we name tRNA synthetases and charged tRNAs. Before beginning with that, let me first explain there are 20 standard amino acids. Okay. Now, standard amino acid means for which we have codons in our genes. So, we have 20 standard amino acids, and because our genetic code is degenerate, one amino acid can be coded by more than one codon. Okay. And this is not always true. There can be two codons or even six codons for one amino acid. Now, if we go by this, roughly there are 41 tRNAs which can bind all the 61 codons representing 20 amino acids. Okay, so 20 amino acids, because of code degeneracy, we have 61 codons coding for 20 amino acids. And how many tRNAs? Roughly 41 tRNAs, which will recognize these 61 codons. Some amino acid, though they can have more than one codon, one single tRNA is enough to bind all the codons representing that particular amino acid. But in all cases, this is not true. Some amino acids who are represented by more than one codons may require more than one tRNA as well. Now, when it comes to tRNA synthetase, the enzyme, we have 20 tRNA, aminoacyl tRNA synthetase. Okay, synthetase enzyme. Each one specific for a particular amino acid. Therefore, we must correctly name them. And let us now learn how do we name 20 tRNA synthetases which are able to identify 41 different tRNAs, each specific for 20 different amino acids. So, if we are going to, like supposing, we will take example of amino acid serine. Okay. Now, if the amino acid is serine, then the tRNA which will bind or accept serine will be labeled as tRNA serine. The first three letters of the cognate amino acid must be written as a superscript on the right hand side. Okay, this is the right hand side, your right hand side, that means. This is the right hand side, superscript SCR. This is how you will label the tRNA which will bind serine. Now, let us understand the name of the enzyme. The enzyme aminoacyl tRNA synthetase. In this case, the name, because the amino acid is serine, this enzyme is specific for serine and its tRNA. The name will become cereal tRNA synthetase. This is enzyme. Okay. Now, when cereal tRNA synthetase enzyme, what is its function? To join, to charge tRNA serine with the amino acid serine. So the product of this enzymatic reaction would be cereal tRNA serine. Amino acid serine attached to tRNA specific for serine. Okay. Hope you understood. If not, let us do it one more time, taking a different example. In this case, let us take amino acid alanine. The cognate tRNA which will bind alanine will be labeled as tRNA alanine. The first three letters ALA for alanine. It will be written as a superscript on the right hand side. So this is the name of what we say the tRNA. Okay. Let me pick up the marker. So this is tRNA alanine. Now, the enzyme which will join alanine to tRNA alanine, the cognate aminoacyl tRNA synthetase, the name will be alanyl tRNA synthetase. Okay. And once this reaction is over, when the synthetase enzyme joins alanine to tRNA alanine, that means it forms a charged tRNA. How it will be called? Charged tRNA will be named as alanyl tRNA alanine. I hope this is now very much clear. Please do practice with two or three more alanine, two or three more amino acid as an example. Let us move forward. So aminoacyl tRNA synthetases are the enzymes which carry out charging reactions or charging of tRNA with amino acids. Now it is simple to imagine that when charging takes place in all the tRNA and amino acids in the same manner, it requires two steps. In the first step, the amino acid is activated. In the second step, it is acylated to tRNA. Then there is an only requirement of one tRNA synthetase, but it is not so. There are 20 specific enzymes, each specific for its amino acid and its cognate tRNA. This tells one thing about this enzyme, that their specificity lies in their structure and the way in which they catalyze their reaction. There are definite reasons why these enzymes are so specific, and why do we have a group of 20 different synthetases for carrying out similar reaction but with 20 different amino acids. Broadly, all the 20 aminoacyl tRNA synthetases are classified into class 1 aminoacyl tRNA synthetases and class 2 aminoacyl tRNA synthetases. These groups differ in the size of active site, the location where active site is located, the structure of the active site, specific motif, number of amino acid, and how they approach their cognate tRNA. Let us understand these differences in some details. The class 1 aminoacyl tRNA synthetase, the active site is 170 amino acids long. It includes the CCA binding domain of the tRNA. That means the active site can bind the CCA end of the tRNA. In case of class 2 enzyme, the active site is quite larger, made up of 250 amino acids, and it does not include CCA binding domain. That means the class 2 active site cannot bind the acceptor arm. The location of active site is also different in these two groups of enzyme. In class 1, active site is located at the C-terminal region, and in class 2, the active site is located at the N-terminal region. Inside the active site, the binding pockets, we can find parallel beta sheet in case of class 1, whereas anti-parallel beta sheets are there in the active site pocket in class 2 enzyme. A conserved domain or fold, you can say structural motif for Rosamond fold, is present in class 1, but this Rosamond fold is absent in class 2. Active site of class 1 enzyme is deep, open pocket, and class 2, it is deep but buried pocket. Now let us discuss how do these enzymes differ in case of amino acid specificity. Class 1 enzymes activates bulky, that means mostly the hydrophobic amino acids such as tyrosine, tryptophan, and phenylalanine. Whereas class 2 enzymes activate polar amino acids which are quite smaller than the hydrophobic amino acids. Class 1 enzyme approaches tRNA acceptor via major groove. Class 2, however, approaches via minor groove. And during the binding behavior, the CCA end deforms during acylation. And in class 2, because the active site does not include CCA end binding domain, the CCA end retains its helicity during the charging reaction. So you can see both class 1 and class 2, they carry out same charging reaction, but structurally they are quite different from each other. Recent research has revealed that aminoacyl tRNA synthetases are not just involved in tRNA charging, but also play roles in disease, immune regulation, and even in synthetic biology, making them one of the most versatile enzyme families in modern biology. So let us now understand some functions of this enzyme beyond protein synthesis or translation. The first thing is that they can have moonlighting functions. Now, what do we mean by moonlighting functions? Some proteins, though their role is to catalyze some enzymatic reactions, but when they are proteolytically cleaved or in their varied different structure under cellular conditions, they can act as some other functional molecule also. This is what we call moonlighting functions. That means the major function is different, but then they can also function in some other form. This is what we mean by moonlighting functions. So aminoacyl tRNA can act as signaling molecule. It can regulate immune responses and inflammation. Now, this is very interesting. It can regulate immune responses and inflammation. It can participate in angiogenesis, that means new blood vessel formation, which is very important when it comes to cancer formation. Okay, so some human aminoacyl tRNA synthetases, their fragments act like cytokines. And you know that cytokines are small molecular weight molecules which can influence cellular communication. So this is what we mean by moonlighting functions of aminoacyl tRNA synthetase. Now the second thing is their link to human diseases. Mutations in tRNA synthetases are now strongly associated with diseases such as neurological disorders, cancer progression, and autoimmune conditions. This is surprising because these are basic enzymes, yet small defects cause very specific diseases. Third is their role in drug targets, especially in antibiotics. Many antibiotics inhibit bacterial aminoacyl tRNA synthetases. Researchers have designed selective inhibitors that target pathogens but not human enzymes. This is important in the fight against antimicrobial resistance or AMR. One of the coolest development is synthetic biology and genetic code expansion. Scientists are engineering aminoacyl tRNA synthetases to attach non-natural amino acids to tRNA. Non-natural amino acids means amino acids whose codons are not found in DNA. This allows creation of proteins with new properties, and the applications are wide, such as we can create designer proteins, novel therapeutics, fluorescent tagging in research. The next property is multi-enzyme complexes in humans. In higher organisms, aminoacyl tRNA synthetases form large multi-synthetase complexes. These complexes help coordinate translation and cellular signaling in much more efficient manner. This shows they are part of bigger regulatory network, not isolated enzymes. Now, tRNA charging is a highly precise and regulated process, crucial for maintaining molecular accuracy. Even a minor error at this stage can have significant consequences. So, how does the cell prevent such errors? Let us move on to the proofreading and error correction mechanisms of the synthetases in our next episode. Till then, thank you for watching.